Hello again, everybody. So this should be the third video of mine that you've watched. At this point, you should have already completed the introductory web quest. You should have also completed the immune response exploration, and you should have completed the ed puzzle, which was labeled 8.5 immune response. All right, in this video, I'm going to review the answers to the immune response exploration with you guys. I'm going to talk a little bit about the ed puzzle that you watched. I'm going to explain the medical application standard and introduce infectious diseases briefly. I'm also going to explain the last two things that you're going to do this week, which are the patient 0 case study and the this podcast will kill you handout. All right, so first I'm going to review this immune response exploration. I know this is a little tricky process. I understand that this might have been confusing to some of you guys, so make sure you're paying attention. Also, make sure to go back and change your answers on your sheet so that they match the answer key if you miss something. So I'm going to read this paragraph again because I think this is really important. So one way in which organisms maintain homeostasis is by detecting foreign cells, pathogens, and particles like pathogens and cancer cells. Once the pathogen is detected and identified, other systems in the organism's body can attack the invader, thus keeping the organism healthier. Cells of the human immune system are finely tuned to recognize and respond quickly to disease-causing agents. So this is the process that keeps you healthy. Things get into our bodies all the time that can cause us to become sick. And this is how our bodies respond to those foreign pathogens. All right, so the first model showing the cell-mediated response, let me review this. The little stars, these are pathogens, so this could be a virus. These guys right here, this is a macrophage, which use phagocytosis to consume the parasite, or the virus, the pathogen. And then it breaks down the pathogen, which is shown right here. And then it takes the pathogen and makes little antigens, which are these spike-looking things right here, and they present them on the outside of their cells. And then these guys, these are the T helper cells, and notice that these are a little bit different. The antigens are a little bit different, the shape of the two-pronged fork. And so each T helper cell is specific to one type of antigen. So what happens, a T helper cell that's specific to that type of antigen on the cell that's infected will bind, and then once it binds, it's going to send out a signal to other T helper cells that are like that T helper cell to produce more. So they're going to start going through mitosis and produce more of those T helper cells. And that signal, that's called cytokines, are the molecules that transmit that signal. Whoops, I just moved everything. All right, so answers. One should be star, so the pathogens are the shape of the star. The diagram number 1 shows that process. Number 3 should be the blob engulfing the pathogen. This guy right here, that's the macrophage. This is called the phagocyte, is what they're calling it on this one. Same thing. Number 4 should be the circle with the fork. I should say like, not link. Whoops. Sorry. The two-pronged fork, that's the antigen, which are the surface proteins. Number 6 should be no, the shape of the fork antigen is different. So if you look closely, they're all a little bit different. Number 7, so in the correct order, D should be first here. So the pathogen is broken down apart by the chemicals in the phagocyte. And then it should be A, the piece of the pathogen is presented on the surface. C, the helper T cell binds to the piece of the pathogen presented. And then B, the helper T cell disperses the chemical signal, which again, those are cytokines, to activate other immune responses. Number 8, do all the helper T cells bind to all antigens? No, not all helper T cells bind to all antigens. Each helper T cell binds to the antigen that specifically matches the surface proteins. So the surface proteins matches the pathogen's antigens, like a lock and key model. It's specific to the shape. Model 2, the humoral response. So this is with B cells. So the previous model, those are T cells. These are B cells, so they're a little bit different than T cells. Notice that the antigens tend to hang out in pairs, but they're all a little bit different from one another. So what happens in B cells, the pathogen, which again is the star here, so this could be a virus, directly binds to the antigen on the B cell, and then the pathogen goes inside of the B cell. The B cell breaks it down using lysosomes, and then same thing as the macrophage, it takes that pathogen, it mixes up the pieces, and it creates its own little antigen right here. So it is an antigen presenting cell, just like the macrophage. So 9, that's called the humoral response. Number 10, yes, the shape of the antigens, the surface proteins, or that look like forks, are all the same in all 5 models. Helper T cells for number 11 bind to the antigens of the antigen presenting cell, and then the B cells bind directly to the pathogens. Number 12, yes, the B cell is an antigen presenting cell. Look at diagram 5. So again, once the B cell breaks down the pathogen, it takes the pathogen and presents it as an antigen. Okay, the last model here, model 3, showing adaptive immunity. Number 13, so what's happening here, the helper T cells bind to the antigens shown on the B cell. So that's right here. So here's the B cell, here's the T cell. They're binding to one another, and then they create these antibodies. So when a B cell is activated by the interaction with the helper T cell, it begins to produce and disperse antibodies, which are those Y-shaped proteins. According to Model 3, what does the antibody do to the pathogens? So the antibodies, this is why they're so important. Antibodies bind to the pathogen, and then the pathogen, the antibodies clumps around the pathogens, so it cannot bind to anything else. So it essentially renders them useless. It makes the pathogens useless. Number 15, the antibodies clumping around the pathogen would decrease the ability of the pathogen to infect its host. So ultimately, killing off all of the pathogens. In Model 4, this graph again is showing over time when we're exposed to a pathogen the first time versus the second time. So a few interpretation questions here. Using Model 4, compare the amount of antibody generated by B cells after the first encounter to the second encounter. So a lot of antibodies are made. 4 times more in the second, encounter. The first encounter is around 20, and the second encounter it's around 80. Number 17, it took less time to reach the peak of antibody production the second time the person encountered or was exposed to the pathogen. The second time we are exposed to a virus, we already have some antibodies against the virus. Very quickly, we make more antibodies against the virus so we don't get sick. And again, it's different depending on every person, right? So if somebody has a compromised immune system, this process doesn't happen as easily as it would in a healthy person. The last question, the booster, so vaccine booster is given to remind the body of antigens of the virus. So some memory B cells. So there are certain types of B cells, they're called memory B cells. They only live temporarily, and they remember the antigens of pathogens. So fun fact, measles, which is a virus that you've all hopefully been vaccinated for, if you get the measles, it completely wipes out all of your memory B cells. So that means you have to go back and get revaccinated for everything, including polio, mumps, everything. You have to get vaccinated again because your body doesn't remember what those pathogens, those antigens on the pathogens are. So you can get sick again from anything that you've already been vaccinated for. All right, so that was the review of the immune response exploration. The Ed puzzle that you guys watched for Wednesday, I'm not going to go through the answers. I'll get in there and give you guys specific feedback, but let me know if you have questions on any of the questions from the Ed puzzle. All right, so now, if you need more help with the cells of the immune system, your immune response, in your guide for this week, for Tuesday, I put a link right here. It says if you need extra practice. Looks like this. This is an applet. It will go through the different cells of the immune system and give you a little bit more review if you feel like you need more review with your immune response. All right, so next up, I'm going to explain the medical application standard, which is right here. So I'm on your Unit X student plan guide. So 3 parts here. So the first thing, what you guys are going to do this for Thursday, describe how emergent diseases spread into human populations, evaluate the effects of social and cultural factors in the transmission and understanding of the disease. So we're going to do the patient 0 case study. And then after that, we're going to look at SARS, which again, I think I said earlier, was it's another coronavirus outbreak. We're going to listen to an excerpt from This Podcast Will Kill You, which you'll do for Friday. And then next week, we're going to come back to this standard, and you guys are going to play a game called Pandemic 2. All right, so I'm going to briefly talk about some stuff you need to know before doing the patient 0 case study. Infectious diseases, one of my favorite topics to talk about. So we need to know what an epidemic is. An epidemic is a disease that's classified, it's classified as an epidemic when there are more cases than expected at a given time. So some rare diseases can be considered an epidemic for very small cases relative to how common that disease usually is. And then epidemics are called outbreaks when they only happen in a certain localized area. So contrasting epidemic to pandemic, when a disease becomes spread globally, it's considered a pandemic when it's a global outbreak. So an example, I'm sure you guys have heard of the 1918 global pandemic caused by the H1N1 virus. It's also known as the Spanish flu. And then the flu, this is influenza A, the H1N1 is estimated to have killed 50 million of the 500 million people globally infected during that outbreak. So very, very deadly disease. We need to know the CDC versus the WHO. So the CDC, that's the Center for Disease Control. They're monitored from the United States in Atlanta, Georgia. And then the World Health Organization is WHO, fills this role on a global scale. So a little bit different. CDC is specific to the United States. WHO is like the CDC of the whole world. The CDC also fills many roles within regulating epidemics in the United States. They produce publications, they track data, they create surveys, they have direct intervention when necessary. And if you go on their website, they actually have a page preparing you for the zombie apocalypse. That's kind of funny. All right, so that's all we need to know for the patient 0 case study. So again, this is going to be linked in Classroom for you guys. 2 things. You will have this PowerPoint that'll act as a guide. Embedded in the PowerPoint, this is the podcast that you will listen to. So make sure that you're only listening to the timestamp for that part. And then your questions, what you will need to answer, are on this handout. So this case study is about the pandemic of typhoid, which happened in the early 1900s. So you need to do this for Thursday. It's just a page and a half, a few prompts. Again, it goes along with this Google Slides. And then for Friday, you guys are going to listen to more excerpts from a different podcast called This Podcast Will Kill You. So this podcast, it's 2 hours long. We're not going to listen to the whole thing. Make sure you're paying attention to the timestamps here again. And you are going to answer a few prompts. So these are really important activities because you guys are going to be collecting evidence for your summative. So make sure that you're answering these prompts because that will help you collect evidence. And then next week we are going to play a game about pandemics, and then that will wrap up our medical application standard. And as always, please, if you have any questions, make sure that you are emailing me.